Good day, everyone. My name is Doaa Salem. I am a postdoc appointee in Biohealth Informatics Department in Ladi School Informatics Computing and Engineering in Indianapolis. Today I am going to present Nm-Nano, a machine learning framework for transcriptome-wide single molecule mapping of 2' O-Methylation sites in Nanopore direct RNA sequencing dataset. The background of this study comes from that 2' O-methylation is one of the most abundant modifications found in both mRNA and non-coding RNAs that contributes to many biological processes such as normal function of transfer RNA and the protection of mRNA against the degradation by the capping and the XO proteins. Detecting Nm modification in RNAs has been a great challenge for many years, and various experimental methods presented in the literature to address this challenge. However, in general, the experimental methods are naturally costly due to the high labor effort involved. So, there have been relatively few computational biology methods proposed in the literature to overcome the limitations of experimental methods for detecting RNA Nm modifications. These computational methods mainly rely on developing machine learning or deep learning classification models to identify Nm sites in RNA sequences based only on short-read data and have not yet been applied to long reads, except for too few research attempts. So the main objective of this study is to develop a framework called Nm-Nano that integrates two different supervised machine learning models, namely XGBoost and random forest with Kimmer embedding models, to identify Nm sites in Nanopore direct RNA sequencing read of two human cell lines, namely HeLa and HEK 293 cell lines. The methodology of this work can be summarized as follows. First, the developed predictors of Nm-Nano integrated in the Nm-Nano framework for identifying Nm sites have been trained and testing using a dataset that comes from one cell line, namely HeLa, in the first attempt, and in the second attempt, the dataset comes from a combination of HeLa and HEK cell lines. In the first attempt, when we trained the classifiers of Nm-Nano on the benchmark dataset of HeLa, we used 80% of the dataset for training the classifiers, and the remaining 20% are used for testing the classifiers' performance. In the second attempt, we use a combination of dataset of HeLa and HEK, and we use 50% of the combination dataset for training the classifier, and the remaining 50% is used for testing the classifier's performance. We should also observe that the dataset that used to train the classifier, either in the first research attempt or in the second attempt, is a bunch of signals that were generated by passing both modified RNA sequences containing Nm sites at known position, identified using the standard Nm-seq experimental method, and the unmodified sequences through the ONT mini-ion device of Nanopore. So the signals extracted by Nanopore device are used to train the classifiers of Nm-Nano. That's it. Thank you.